Dr. Chris Flowers, thank you very much for taking the time, sir. I know you're quite busy. I want to have you talk about this really kind of bombshell report that your team has released, and I'll preface my first question by saying this: a couple of years ago, when Joe Biden got the injection, there was a huge to-do about it. All the news channels covered it, and they broke into regular programming, and Joe Biden is getting the vaccine. And I remember sitting there watching this, and I don't think I've said this on the air, but I remember sitting there watching this, and I was thinking to myself, there is zero chance that what's being injected into this man's arm is what's being injected into the rest of our arms, because they just couldn't afford to have this man keel over and drop dead or suffer any adverse reaction after getting this injection. Now, what you guys have uncovered is not exactly that, but is similar in some ways. Tell us what you guys have found.
Well, a lot of the work has been out for a little while now. That's this issue about process 2. It's the new form of manufacture. So there's Josh Groskow and Kevin McKiernan, for example, he was sequencing the vaccine leftovers and found all these DNA plasmid fragments contaminating what's supposed to be a clean mRNA vaccine. And so that has been noticed for quite some time. The important thing from our point of view is the work that we did based on some of the initial work on the data, trying to identify some of the people who received this in the clinical trial, because it turns out the Pfizer clinical trial had multiple amendments. Now, what happens with a clinical trial normally? You write a clinical trial, and the protocol has to be approved by ethics committees, and then it has to go past. In this case, goes past the FDA to be approved.
Right.
So what happens then? The research protocol is locked in place. You can't change it until now you finish the trial and analyze the data. And if you want to do something different, then you run a second clinical trial.
Right.
Now, the Pfizer clinical trial was extremely different in that there was a multitude in the teens of amendments. And around early October of 2020, there was a request, and it was actually in the protocol, that this process 2 would be tested on approximately 250 subjects. But because they keep doing these protocol amendments, they almost immediately issued another protocol amendment, and it hid some of the newsworthy stuff, as it were, about this process 2. And most people didn't really know about it for a long time. So that was the first bit. So for people who haven't heard perhaps about process 2, process 2 is the Pfizer's name for the new manufacturing technique they were using to scale up the vaccine production. Because the way it was done initially for the clinical trial, BioNTech in Germany had been manufacturing the mRNA, and the lipid nanoparticles were being processed for them using a technique very similar to the amplification techniques that are used when you remember having the swab thrust up your nose and tweaked around.
Right.
That uses something called PCR. It's polymerase chain reactions. Basically, it's just amplifying what's there, and they keep amplifying it. So they talked about how many cycles they were doing, and they had so many cycles they could basically pick up DNA anywhere.
Right.
And they were picking up the COVID that way.
And this was reported by the New York Times, no less. So this is not conspiracy theory.
No, it's not. This is not, because this is everything I'm telling you basically is from the Pfizer documents. None of this sort of stuff is made up. But what happened is they used the PCR to actually produce the amount of vaccine they required for the clinical trial. But it was very expensive, number one. Number two, it was slow, and they couldn't produce enough of it. So what they did was in the interim, while the trial was going on, they were ramping up the manufacture using a very commonly used technique, which is the use of E. coli, that's gut bacteria, basically, to take a little piece of RNA, which is wrapped into a circle. It's called a DNA plasmid. And then when that gets into the E. coli, the E. coli just, like a photocopier, bang, 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 produces lots of it. So the end process of manufacture is to destroy the E. coli cell walls, let all this stuff out, and then clean it up and harvest the mRNA, which is what they were wanting to inject into us. Now, the problem was, from the data that we've seen, is that this wasn't really cleaned up properly. And if Pfizer would come out quite early on and said, "Oh, this is a bit dirty, but we promise to, we're open and transparent. We're in the process of cleaning this up so that there's very little contamination." But there are concerns, and you may have come across Professor Buckholt's testimony in the South Carolina COVID inquiry. Yes. And he's a cancer genomics person. Yes. Genomics is at the study of the genome, the genes, and in particular for him, it's cancer genes. Now, of course, I'm very interested in that, being a cancer radiologist and having been involved in breast cancer diagnosis, treatment, and many clinical trials over 40 plus years.
Right.
So I have a lot of experience in this area. And he in particular was very concerned about the degree of this DNA plasmid contamination, and because they're fragments, and the fact that they're not just around the cell in the bloodstream, they actually get inside the cell. Now, why on earth is that important? Well, if it's in the blood, your enzymes will come along and just recognize them as abnormal, and they'll eat them up. There's those white cells called macrophages that come along. They gobble up foreign material, if you like.
Right.
But with the advent of lipid nanoparticles, they actually inject all of this stuff into the cell. Once these pieces of RNA are inside the cell where they're not supposed to be, they're very close to the nucleus. And all it takes is for the nucleus of that cell to divide, because the cells are growing. Then there's an increased risk of any of these fragments incorporating themselves into your genetics, your own genome. And that has always been the theoretical worry. And especially since we've seen the advent of turbo cancers and things like that.
Yes.
And the dysregulation, the unregulated growth of cancers, things like this. These are things that make us very concerned that there's a link between the vaccine, the contaminated vaccine in particular, and these cancers.
So what you guys have found, if I'm understanding correctly, is that this process 2, this more contaminated process, this is different than the process 1 that Pfizer kind of said, that Pfizer tested. Hey, we're using process 1 to run this clinical trial. And then when it came time to ramp up mass production, they used this more contaminated process 2. Is that correct?
That is correct. That's well known. It's been published by many people now over the last few months. So people are indeed very, very concerned.
Now, does this change in process materially alter the solution, whatever you want to call it, the injection that's being put into people's arms?
Well, unfortunately, it does, because when you compare this new vaccine manufacture, and I use vaccine in the new FDA definition of a vaccine, because it doesn't fulfill the old vaccine terminology.
Good point.
What it does is really quite significant, because without the contamination, you've got some adverse events, but they're relatively small compared to what you got in the process 2 people. And the trial patients, they only let me roll a little bit further back. If you remember, I was talking about protocol amendments.
Yes.
They the Pfizer, or should we say BioNTech in particular, because it was at that stage was being overseen by BioNTech, who was taken over by Pfizer. They were running it in Germany and manufacturing it, and they basically wanted this to have a trial on it, which is good and worthy, but they only wanted it in 250 people, not 44,000 people were originally scheduled to be in the Pfizer clinical trial.
Right.
So this was the idea of doing this was introduced in one of the protocol amendments. That was Protocol Amendment 6 on the 6th of October of 2020. Now, one other team called OpenVate actually found the 250 and handed that spreadsheet of those study numbers over to us. We were able to clean up that data, identify them because of the batch data they'd passed on to us. We were able to identify the 250. It was actually 252 people who received this new process. We also found the placebos. That's the people they were being studied against. We were able to identify they were done in 4 different sites in the US. None were overseas, all in the US. And they were done over a very, very short period, just before the EUA. That's the Emergency Use Authorization that was granted by the FDA in the middle of November. And these 250 patients, one in each arm, that means the placebos and 252 of the people who received this new process too. They were done within a short space of time, as I say, and separate from the regular part of the clinical trial. So it was quite clear that this was a distinct, on its own, clinical trial.
This was a trial within a trial, in effect.
It is, and we've entitled it, a secret trial, but in reality, it's not actually secret because it was in the protocol amendments. But the way they produce these amendments means that you don't know about it, and it's never really been publicized that this was going on. They didn't tell us that was only 250 people were testing this before it went, that everybody received it in their arm, in Europe, in the US, around the world. If they received the BNT 162b2 in the US, or Comirnaty, which is the commercial version of that the rest of the world got. So it was a bit of a problem, because going back to the ordinary routine clinical trials, you don't change anything about the drug that you're testing during a trial. You've got to finish that trial, and if you want to change something, then you can repeat the same trial, but with this new substance, because that's basically what it is. It was different. And so when we came around to analyzing these 250 patients, we showed that there were 2.4 times more adverse events in the people who received this vaccine, which is tainted, if you like, with the DNA plasmid fragments, compared with the placebo group, who are supposed to receive just saline injection in the arm.
So this product produced by this process too is 2.5 times more harmful than.
Yes, and I think we've talked about serious adverse events in the past, and when people are talking about that, they're talking about the evidence that's come out of VAERS that was reported in the post-marketing experience by Pfizer, which was the first 3 months of the clinical rollout. And then we had corroboration of that data in the European periodic safety update reports that came out every 6 months. And those were on huge numbers, 17 million plus people, or 1.3, I think was the first one, 1.3 million people who'd been vaccinated in that 6-month period. And we saw the sheer amount of serious adverse events that were occurring. And it mirrored all the findings we found in the original Pfizer documents. So again, it's corroborative evidence. But everyone really who received the serious adverse events, they were receiving the new process 2, not process 1 that the trial was based on. So for that reason, we believe this is serious, this is significant enough to to warrant serious inquiry, questions being asked in Congress, and really the FDA being pressed as to, why did you incorporate this in the trial? Why wasn't it separate? What about these serious adverse events in this type compared to the other? What about the contamination? What are you doing to clear it up? It's all the usual things you'd expect your government to be following up on and trying to improve for the population, if they're not distracted by lots of shiny objects everywhere right now.
Well, you said it. You've alluded to this a couple of times already, but I just want to emphasize, is any of this normal best practice?
Absolutely not. None of the manufacturing process is best practice of the Pfizer vaccine, because they actually put it in their contracts. With each country, there are several contracts that have either been leaked or FOIA'd, and they all have exactly the same paragraph in it, which talks about that these products are not to be serialized, which is part of the good manufacturing and good distribution process, of which there are manuals for the EU and the US. It's supposed to be highly regulated. They're supposed to make sure the medicines we receive are clean. They're not likely to be toxic and cause us problems. But here we go.
Did these 252 people that you've identified, did they know that this was being injected into them?
We don't know for sure, because we don't know. There's no informed consent forms available for these people. We don't know if they had a different informed consent or whether they were just told they were receiving the vaccine. We just don't. There is no evidence. We have no idea. And these are very good questions, Mike, and things that should be asked.
I mean, I'm starting to get I've had this idea for quite a while, but you're starting to get the picture of why they wanted these documents hidden for 75 years, right?
Indeed. Mm-hmm.
But this has to be illegal on some level. I mean, the least of which the Nuremberg Code comes to mind.
Yes, and I don't know if you've heard or watched any of the European Union COVID inquiries that they've really delved into this and pushed the Pfizer executives about their manufacturing processes and the contamination. Now, they have been asking questions in Europe, which is, I think, more why they're reluctant to give more of the vaccine, which is quite nice in the way it's hitting Pfizer's bottom line. But at least they've been asking questions and wanting to know the answers. We don't have that same sort of questioning here. For some reason, the press aren't interested at all. If you question anything about the vaccine or the outcomes from the vaccine, you're just basically labeled as a conspiracy theorist. But this is data. All we're doing is we're asking questions. We want the answers. Let's have some clear answers, and let's have a safe medicine to use.
Do people who are subjected to this this highly irregular, and I'm being charitable there, this highly irregular experimentation, do they have any recourse if they find out this was done to them?
Again, we don't really know. We do know that the PrEP Act basically protects big pharma from the vaccine point of view, protects them from any form of litigation. We are advised that if we can find evidence of fraud, basically you can build a RICO case against big pharma for that, but you need a lot of evidence. Now, we found small pieces of evidence, but individually, how can a small team go up against it's like David and Goliath, but this is a much bigger giant. It's like the key, I think, is going to be if I think is ICANN, I think, or one of the other teams who have actually brought an amendment. They're trying to amend or get a decision from the courts on the constitutionality of the PrEP Act, because it really deprives people of the normal protections of recourse for injury. They've invaded your property, which is your body.
Right.
They've caused problems.
Right.
You want to have some form of recourse, and that should be constitutional. But because of the PrEP Act, that is not a recourse that you were actually able to have.
No pun intended, Doctor, because you're part of the DailyClout WarRoom team. But WarRoom this for me. How is Big Pharma going to explain this away? First of all, is there any chance that this was a mistake?
Well, I think a lot of the times we can put these things down to incompetency.
Right.
Don't forget these are giant companies, and they often have minions working for them who really don't know what much of what they're doing. They just do as they're told.
Right.
And there was the pressure of warp speed and the fact that money was flowing, and the government wanted a product immediately.
Right.
So there's all of those things. So one could attribute that just to foolish mistakes and oversight, lack of oversight, all these sort of basic things.
Right.
But you'd expect at least the arms of government regulation who are supposed to protect the people from rogue vaccines to give us some form of protection. And the way the VRB PAC and AICP have performed as regulators in this instance, and really the discussion every time we watch these 6 monthly meetings, or more frequently sometimes, approving vaccines, they just it's like a rubber stamp process.
Right.
And they basically take what they're told as gospel without looking at the data themselves. So when you've done clinical trials so long, even as a primary investigator, you look at your own data. You want to know, have I done something? Am I doing everything absolutely right? Because back in the day, I keep saying back in the day, but because I've been involved with clinical trials for so many decades,
right.
I remember what it was like going to the FDA in Silver Springs and having to produce all this documentation and proof that you weren't going to cause any type of adverse event.
So there was a time, Doctor, where this agency actually worked for us. There was a time.
Exactly. That's what I'm saying. I'm a bit too old school, I guess. That's what makes me angry. Yes. Is that it's not what it used to be, and they used to protect us. And my overall feeling is that these committee boys are not protecting us. They've got lots of connections with the company they're actually regulating.
Yes.
And then, of course, they're only on the committee for a short time, and then there's this rotation out to Big Pharma, like you saw in the UK with one of the public health chiefs. That was Jonathan Van Tam, who was on with Boris Johnson every day during the pandemic, talking about getting people to take this vaccine. And now he's partially given up his job at Nottingham University in public health, and he's being paid by Moderna. So I don't know if you'd heard that, but
I had not. But it doesn't surprise me. The revolving door is very, very real.
Yeah, it was big news when I left the UK to come back to the States a few weeks back. Yeah.
I remember the question that stumped me a moment ago. Is it a stretch to say that there are different versions of this vaccine, or is it more accurate to say that there were just different manufacturing processes? Because it seems to me that the different manufacturing processes gives rise to a different version. Is that a stretch or no?
Well, that is how it should have been treated. As I say, that's why it shouldn't have been 250 patients. It was only a couple of weeks before they gave approval with the EUA. But they should have said, right, now let's do the same thing. It only took 2 months. Let's take another 2 months and let's look at your new product, because it is different and it should have been checked, and it wasn't checked. So we were walking into no data whatsoever, and basically the people who received the vaccine in the first 3 to 6 months are the ones who've basically been the guinea pigs, and the post-marketing experience, the follow-up with all the serious adverse events, is the evidence that should be looked at back on and said, perhaps we shouldn't have done this way.
Now, did the FDA, just to circle back to the FDA, are they aware of this? Were they aware of this? Is there evidence to show what they knew
Well,
and when they
it
knew
was
it?
in the FDA Pfizer documents. So the question is, how much did they know and when did they know it? But
Right.
we knew they published on October the 6th that they were about to do this process. And normally, when you're doing something like this, especially at the end of a trial, and you want to try and approve a vaccine quickly for the public good,
Mm.
what you're mainly concerned about are any adverse events. Because the first thing is, does it provide an immune response? And that was the only thing they basically said they wanted. They wanted to have a product that produced these antibody titers. That means strength of antibody in your blood.
Mm.
But normally they would be looking at adverse events as well. And in the past, when serious adverse events have occurred during a trial and anything else,
Mm.
the trial has been stopped. I mean, this is true of the swine flu vaccine in the 1970s,
Mm.
where there was only a couple of Guillain-Barré, I think it was Guillain-Barré that they had, and they were withdrawn. And yet Guillain-Barré is a very common serious adverse event from the Pfizer vaccine.
Right. I remember very early on in this pandemic, we heard a lot about the cytokine storms. And I heard you mention a connection between these findings and that phenomenon. Can you tell us a little bit about that?
Yeah, so a cytokine storm basically is when it's like an avalanche, really. You get an antibody being formed, and then suddenly you get much more and more of a response, and that generates a bigger response. When you've got stiff lungs, which is basically what happens when you actually get COVID, a cytokine response is a horrible thing to have, because it basically means that you drown in your own secretions, number one. Number two, ventilation obviously isn't going to work, because you can't get oxygen into someone who's got no capacity to be able to absorb the oxygen, because all the lung surfaces are full of this debris.
Right.
And so they tried things like putting people on lying on their stomachs and try and ventilating them upside down, if you remember.
Right.
That was one method. But basically, once you got on a ventilator during COVID, that was a sort of death sentence.
Right.
Effectively, you really didn't want to get to that stage. But there is a very interesting connection between the vaccine, lung cancer, and cytokine storm, which I'd like to share with you from a paper. It's a paper that came out about 4 weeks ago, and it was looking at patients that they were treating with lung cancer, and they also traced. They had they had records of the vaccine they had, when they had it, the dose and everything else, and then the occurrence of any adverse events after that. And they found that not only did these lung cancer patients have issues, they actually had this cytokine storm as an effect of the vaccine. Now, whether this potentially, in theory, could be because of the one side effect of the COVID vaccine was COVID itself.
Right.
It's like, back ages ago when they started with polio, they started with a live polio virus, and some people got polio.
Right.
So they attenuated it, they heat treated it and made it less infectious. But even so, some people still got polio.
Right.
Same with the COVID vaccine. There is a group of people who got COVID from having the COVID vaccine, and that would be one explanation for it.
Right.
But the other explanation is this issue of the vaccine affecting the immune system. And you may have interviewed people, Dr. Makis, for example, when we're talking about the shift of IgG1 to IgG4 in cancer patients in particular. So that's why you get more of the hematological malignancies just turning up, leukemia, lymphoma, because you've changed from your normal white cells that recognize foreign material and attack it to IgG4, which is the mechanism by which it recognizes this is part of your body, don't attack it.
It attacks itself, right?
It acts as a protective mechanism.
Yeah.
And it's not only that, you see, we get down regulation of some genes and up regulation of others. And these are very important when it comes to cancer, because it's the it's these genes that control cellular growth that we attack with our chemotherapies. That's how we deal with it. There's all sorts of different names for the various therapies that you can use in the different types of cancers. That's thanks to the advances of the last 10, 20 years, really. But this is, when it's occurring as a result of a vaccine, then you have to take note and think deeply, what on earth is happening? Why is this happening? And also, what can you do about it? And I don't have any answers now. I'm still waiting to see if anyone comes up with an answer to that, because I've not seen any good explanation of why it causes that cytokine storm, or what is the mechanism by which they get deregulated. Could it be the integration of some of that rogue contaminated RNA fragments into your own genome, or is it simply some other mechanism? We don't have that answer. And yet this is not a phenomenon that's really restricted to the US. I did an interview on Croatian TV with the professor of pathology at University Hospital of Zagreb a few weeks back. We're discussing turbo cancers, and she shared with me the data that she'd observed, because she'd collected all sorts of data on cancer patients before then. So she had a fantastic way of showing this is what it was like before the vaccine. And then after the vaccine, I suddenly started noticing more cancers rapidly growing, people coming in and dying very quickly from their cancers and things like that. So she was able to show from her own database in Croatia the effects that we've observed in the rest of Europe, the UK, and also in the US and Canada. So it's a worldwide phenomenon, and something that people should be asking questions all the time. Why aren't people in Congress keep questioning? Why aren't the media asking questions like this?
I mean, this should be, this report that you guys put out and these questions that you're raising, this should be front page news of every publication, not just in the US, but in the world. It should be the top story on all the major broadcasts. I mean, this rolls right into my next question on turbo cancers. How prevalent of a problem has this become in recent weeks and months?
Well, it's bothersome enough. I think the answer is it's still relatively small numbers, but they are significantly more than they used to be. They're a factor of 2 to 3 times at least, based on the Zagreb data, where we've got a linear table of what happens over a period of time.
Have turbo cancers been with us for a long time, or are they new?
No, first of all, turbo cancer is a term coined by popular demand, as it were. This was crowdsourced turbo cancer, but it explains exactly what is happening, because you've got cancers, occurring in someone who was totally well, had normal scans and everything yesterday. Now they a week or two later, or even following day, they're diagnosed with a tumor into hospital. The next day, after they've died, with metastases everywhere around the body. I can tell you, I've seen hundreds of thousands of cancer patients, and yes, you see it occasionally. I've seen 2 or 3 in my lifetime. So for a pathologist sitting there in a lab with all these cancers suddenly coming across her desk, and at least she was asking questions and she put 2 and 2 together. She has no political axe to grind or anything. She's just a regular doctor doing her business.
She is, dare I say it, following the science.
She's a proper scientist. She tried to present this data in front of experts at her hospital and a regional meeting, and they listened to her in stony silence and didn't ask any questions. Which, if you've ever been to a medical meeting, you normally get people try and pull you to pieces and ask questions all the time, but this lot were silent. And they were, these are the top experts in their country. So it does make you wonder, what has happened. There is an issue about the lack of critical thinking. It's in virtually every profession now. Everything, including, maybe that explains the media to some extent.
I
Why
think
aren't
you're absolutely right.
you to do?
I mean, it's just been completely the scientific method, for example, has been completely inverted. Well, in the few minutes that we have left here, Dr. Flowers, I want you to tell the folks about your great team at DailyClout, what you guys are doing, what you guys are up to, and where people can find your great work, because you're doing the work, unfortunately, that these agencies that have been chartered to protect us, they're just not doing it, and they have no interest in it. And it's up to great people like yourself and your teammates who are doing the work they should be doing, but sadly are not.
Thank you, Mike. Well, I'm just a representative of the team. I'm not anything special. I'm just a regular doctor who's been involved in trials and wanted to help out, trying to find out what happened in the last few years. We have 3,500 volunteers who started on this journey with us, which include a group of attorneys working pro bono and working with some of the AGs to get some lawsuits underway. The rest of us are basically plowing through the Pfizer documents, and virtually every month we get another addition to the Pfizer data dumps, and basically we're still missing pieces of the jigsaw. And these are the very interesting pieces of the jigsaw. We know what we're looking for, what we're waiting for, because that is a particular shape. There's a particular piece of data missing, and there has been a lot of data released even in the last dump that is going to be subject of another report. Or you can find everything that we do on dailyclout.io, and I also have a Substack called drchrisflowersmd.substack.com. You can find me also on Getter at Chris Flowers MD. But if you go to the DailyClout, if you click on Pfizer reports, then all our reports that we have published online are there. If you'd like to, you can receive a copy of the hard copy of the best-selling first 40 reports that have been produced. We self-publish those, available for people who like a physical copy that they can share with their relatives, put on the coffee table, even as a talking point. Right. A conversation starter.
Right.
But I would tell you that if any of your viewers can help out with supporting DailyClout, because all of the work that we do, we're doing it for free, but a lot of people in the background, there's the sort of background things that go on, including running the organization, need supporting, and with all the lawsuits that are going on, we need assistance from small donors like you and I for this work to continue. And of course, now we're beginning to get the Moderna data, and we're having the children's ones right now. And later in 2 years' time, we're going to have the similar sort of data that Pfizer produced for us. We're ready to go. The teams are ready to go. They love your support. So please, if you see them on Getter talking, or Twitter talking about the reports, please give them a thumbs up, a like, repost, quote tweet, whatever you like to do, comment on it, share it with your friends. That's the most important thing, because if we raise awareness amongst the population, one day this small wave will turn into a tsunami.
Very well said. I couldn't have said it better myself. Thank you, Dr. Flowers, for taking the time, and thank you for putting the time and the effort into putting these pieces of this puzzle together, sir.
You're welcome.